Hi, everybody. Pardon to the people who are behind me. I hope you're here for the State of Neuroscience. My name is Mariette DiChristina. I'm the editor-in-chief of Scientific American, and I can promise you, since I've had a chance to get to know the speakers a tiny bit here, that you're in for a rare treat of insights about something that is so well known to us in so many ways, since we each have one, the brains inside our skulls, but also so mysterious, and yet under a lot of scrutiny, and really valuable scrutiny, by the research community. I'm the editor-in-chief of Scientific American, as I've mentioned, which was started in 1845, when the idea of being a scientific person was really aspirational. And also wanted to mention that we have a publication called Scientific American Mind, begun in 2002, which I hope you will also check out. So I'm going to briefly introduce the panelists around me, and then we're going to begin a conversation. I wanted to let you know, too, that I will be keeping an eye out and giving you plenty of opportunities through our conversation to ask your own questions. And I hope that through this, we'll have an exchange of ideas around the brain, around things we wonder about, around things that we are coming to know increasingly well. And I'm really looking forward to learning from it. So in order, in front of me here is Nicola Allen, who is assistant professor at the Salk Institute for Biological Studies. My side here, Ronen Gadot, CEO of ElMindA in Israel. Michael Hausser, across to my left, a Wellcome Trust Principal Research Fellow and Professor of Neuroscience at the University College London in the United Kingdom. Weidong, Professor of BioX Institutes, Shanghai Jiao University, People's Republic of China. And Hideyuki Okano, a Professor of Physiology at Keio University in Japan. Welcome. And thank you very much. I think it would be really useful if we could kind of start at the large scale about the brain initiatives. And so, Professor Okano, could I ask you to speak a bit about what Japan is doing? There are something like a half a dozen large brain initiatives around the world, and they're doing many kinds of exploration of the brain. Could you talk to us a little about what you're doing?
Let me tell you the situation of the brain initiative across the world. Okay. So actually, April in 2016, so worldwide leaders of the G7 country get together and made a statement about object of the brain initiative. That include fundamental research with international collaboration. And second is a global program for diagnosis and prevention and treatment in brain disorders. And third, theoretical modeling of the brain and development of the brain-based artificial intelligence, AI. And fourth is integration neuroscience with social and behavioral science to improve education and the management components of the brain-aware societies. Actually, there's a growing the number of the brain mapping projects started in many countries. In 2013, US started a brain initiative, and Europe started a human brain project. As regards human brain project, it is focusing information and communicating technology platform, and so they are more dedicated to the cognitive neuroscience and also theory of how brain works and information processing. And so, but U.S. brain initiative is more such a bottom-up type. That is investigator-driven science that is going to also include developing the innovative technology for the neuroscience. Actually, so in Japan, we started its own brain mapping project named BrainMinds. BrainMinds is more focusing on a small neural primate called a common marmoset. We are going to investigate the network of the common marmoset in order to get insight from the information processing and disease in the humans as using marmoset as a model. And also, there's also emerging tendency of the brain mapping project across the world. In China, officially not studied yet, but the project is already approved by the central government, formally named Brain Science and Brain-like Intelligence Technologies. Moomin Poo in Shanghai is one of the leader for it. And China Brain project is characterized the using macaque monkeys as a model. And also, in my understanding, Korea and Israel and Australia will start their own brain mapping project. So international collaboration is very important to elucidate how, in order to understand how our human brain works and how to understand diseases.
Could I ask just one follow-up? You mentioned about using the marmosets, and could you speak just a little bit about the value of using the non-human primate in your research?
Okay. Yeah, because non-human primate is well-developed the prefrontal cortex that is involved in very higher brain functions, and so that is those cerebral cortex is expanded during evolution after they become a primate. So they have some primate-specific function. Those are often the target of the various mental disorders. So in order to mental disorders, understanding primate brain is very important. And also there's a tendency of the genetically modified non-human primate came to be possible. So actually our group, in collaboration with Erika Sasaki, we developed a lentivirus-mediated gene transfer, making transgenic marmoset, and also genome editing. And also the Chinese group, and they are successful in making the knockout macaque monkeys by using genome editing. This was enormously contributed the genetic basis of the neural circuit and in the primate, even the prefrontal cortex, and also understanding human diseases.
Thank you. Li Weidong, if I could come to you for the perspective from the People's Republic of China.
Yeah, so like Professor Okano mentioned, so from 2013, European, US, and also Japan started their brain project. Then at the time, the Chinese neuroscientists also want to do some contribution for the whole field for related neuroscience. So there is numerous conference happened in China, but after 3 years, we haven't started yet. But after the very deeply discussion between the hundreds major neuroscientists, so at the end, there we came to the common ideas, which is build one scheme, one body, two wings. The one body is like other countries doing, so the basic neuroscience. We want to investigate the principle of brain function and like how to build memory, how to produce the language and so on. And also, in addition to this main body, there's two wings. One is related to the disease, the brain disease. Brain disease including two major parts, neurological disease like Alzheimer, Parkinson, and so on. And another part of brain disease is a psychiatric disorder, including schizophrenia, autism, depression, such major mental disorder. And another wing of China brain project scheme is the brain spread new technology related to computation and also artificial intelligence. So this is a very ambitious goal of the China brain project. And also for the main body of the China brain project, there are also Chinese neuroscientists want to develop some new technology to help the progress of neuroscience. Like there's the team in China, they made this great contribution to do this micro-optical sectioning topography led by one amazing scientist. So they can cut the mouse brain or the primate brain automatically. Then after a few days or a few months, then they can rebuild the whole neural connection in the whole 3D whole brain. So it's amazing progress for the neuroscientists to investigate the mystery of brain. And also, I think China can contribute for the whole world and humankind. It's because China got help for the whole countries all over the world. So the economy booming in China, we have a lot of funding. Then we recruit more excellent scientists from all over the world, including Dr. Moomin Poo, returned to China from US after he established an amazing science career. And this talent guy return to China, work along with this local Chinese neuroscientists like the fellow for Chinese Academy of Science, Dr. Yang Xunli, Dr. He Lin, Dr. Duan Shuming, Dr. Zhang Xu, and so on, along with the young scientists. So I think China can contribute a lot because the funding, because the talent. And also another contribution China can do for the whole world is China has a great population. So for the brain disease, all of them are very complex. So we need like a huge sample number. Then you can deal with the real cause factor for something like schizophrenia or depression. This affects a lot of people's life. And China, we're doing well in this field also. Thank you.
Thank you. Professor Hausser, this has me to mind of thinking, so there are many large projects we're learning about, approaching the brain in various ways. How are we going to begin to integrate these learnings? What can we do there?
Yeah, that's a great question, Mariette. I want to start by echoing what Professor Okano and Professor Lee have said, which is that this is really an incredibly exciting time to be working in neuroscience. And we have some amazing new tools for interacting with the brain. First, we're entering a new era of large-scale recordings from the brain involving the use of new microscopes and new generation silicon probes that allow us to record from thousands of neurons at the same time in the awake brain. This is orders of magnitude more neurons that's been possible than has been possible previously. And second, we also have new, precise, and very powerful means of manipulating brain function, such as optogenetics, which is a new approach of using light to control neurons with millisecond precision. Now, at the same time, it's becoming clear that the conventional model of neuroscience research, of scientists working independently in their own labs, is inefficient and might not be up to the task of solving how the brain works. First, many experimental labs are collecting terabytes of data per day, which far outstrips the capacity of a single lab to analyze and process this data efficiently. And secondly, we still lack a general theory of how thousands of neurons across many brain areas interact together to control behavior. And so I'm part of a new grassroots collaboration, a new initiative called the International Brain Laboratory, which aims to create a new model for collaboration in neuroscience. So this is a virtual laboratory that's linking together 20 experimental and theoretical labs across the world to share data and develop a unified theory of how different brain areas work together to drive behavior. And with this initiative, we hope to provide a template for a new kind of collaboration in neuroscience that can change the culture of neuroscience research.
Thank you. So far we're talking a lot about neurons. That makes me wonder, are we doing enough to get a complete picture of the brain?
Yeah, so I think as my colleagues have already talked about, we have a lot of challenges in neuroscience right now about understanding how the neurons in the brain communicate with each other, how they normally function, and how they might be going wrong in different neurological disorders. But I think what's missing from the conversation so far is that we haven't really talked about the rest of the brain. So as many of you probably realize, neurons are only half of the cells in your brain. The other half are actually a group of cells called glia. And essentially, glia means anything present in your brain that isn't a neuron. And we think that these cells could actually be having a very important role in determining how your brain normally forms, how it changes throughout life, and could actually be going wrong in different diseases. So when we talk about glia, we're kind of talking about a catch-all term. So there's three main types that people think about. One's called an oligodendrocyte, and they actually act as an insulator on the neurons and allow the electrical signaling to be quick. And actually, these are the cells that die in multiple sclerosis, so you can see how important they are. Another type is called an astrocyte, and these are star cells, star-shaped cells that touch all of your neurons, all of their connections, and they're actively involved in responding to neuronal transmission and releasing their own chemical messages to affect neuronal communication. So these cells are also kind of, we should be thinking about them as an active part of the neuronal circuit. The third type, which has had a lot of attention recently, are called microglia. And these are actually part of your body's immune system. So they're present in the brain in order to fight off infection and also to eat up debris and keep the brain healthy. But we know from recent work that perhaps in many different neurological disorders, these cells are too active, and they could be eating up too many neuronal connections and contributing to a lot of the damage. So I think what we think about and what I encourage in the discussion today is to consider all the cells in the brain and the whole brain and thinking how they work together and interact in order to kind of make some progress in thinking about these big challenges.
Thank you. So obviously when the brain is interacting, there's an activity that's going on. There's blood flow, electrical activity. You and I were talking a bit about electrophysiology and how that can be used to study patients along the way and see how things might be developing, but also potentially therapeutically. Could you speak to that a bit?
Yes. To touch upon electrophysiology or the circuitry of the brain, as entrepreneurs, we try to take a very practical approach. And we know that there's still a lot to learn about the brain and there's a lot of unknowns, but still there are 1.5 or 2 billion people that suffer from brain-related disorders. And we need to see if we can do something quicker than, until we learn a lot about the brain. And we look at those patients. One of the common threads is that the current available tools, imaging tools and other diagnostic tools like CAT scans, MRIs, PET, diffuse tensor imaging and such, are great tools to provide us information about the structure of the brain, about the metabolism of the brain, about how the brain is using energy, how blood flows, but still to figure out the function of the brain, things like attention, like memory, like depression, like dementia, and like pain, we still have to heavily rely on clinical symptoms and behavior. And of course, the gap is because the brain is extremely complex, and it comprises of these 80+ billion neurons and glia and other stuff that are connected in 1 trillion connections. So every second that I speak and everybody is listening and we think and we feel and we process information from around us, there are hundreds of millions of neuronal firing inside our head. And unfortunately, there's no good way to measure those neuronal activities non-invasively. Of course, there are better tools to measure it non-invasively, so invasively, I mean. So one of the, I think there is a surge of additional research, and the Brain Initiative is also speaking about it a lot, is try to better understand the electrocircuitry of the brain and how the dynamics, or how, so to say, the flow or traffic along those neuronal connections are creating functions, and how this relates to structure, how this relates to metabolism, how this relates to behavior. So when you talk about electrophysiology, is an area that my company is also heavily involved for the last 11 years. We look at 3 layers that some of the initiatives are trying to figure out. First is sensors, how to measure electrical activity or neuronal firing. So there are invasive ways to do that, and there are non-invasive ways. The non-invasive ways are not great. They are measuring the electrical field around our head or the magnetic field and trying to figure out what's going on inside. But there's a lot of development in improving those sensors, both implantable sensors as well as external sensors. The second one is analyzing the data. After you collect the data, how you analyze the data and correlate it, or see how this data of neuronal firing relates to structure, relates to metabolism, relates to glia, relates to behavior, and try to find predictors or patterns that are indicative of specific diseases. So the analysis is a major part, and I will discuss it later as well, because this requires a lot of data and analysis techniques in order to figure out from the data. The last part, which also Professor Hausser touched upon, is manipulating the electrical circuitry of the brain. So we see also a lot of research into how we can manipulate and neuromodulate the circuitry in order to create change, either to treat disease or to enhance our capabilities. So there are various ways to do neuromodulation. Deep brain stimulation is the let's say, the most common, that already 100,000 people with implanted deep brain stimulation exist in the world with Parkinson's disease. Very effective in Parkinson's disease. A lot of research how to bring deep brain stimulation for additional diseases like obsessive compulsive disorder, depression, but movement disorder is the most common. Other neurostimulation techniques are non-invasive from the outside, trying to manipulate the neural circuitry with either magnetic field, with what is known as TMS, transcranial magnetic stimulation, or electrical field, with either direct current or alternating current or random. And we see also some nice advancements in development, the recent one coming from Ed Boyden's lab, to be able to show how we can do deep brain stimulation actually from the outside with electrical activity from the outside. So these three layers are very important, I believe, to be able to provide maybe quicker solution. It's not the perfect solutions, but to address clinical needs, and while addressing, of course, learning more about the brain. So we are trying to look at what can we do in the next, let's say, less than a decade in order to really alleviate the issue of brain-related disorders, trial and error, which to a large extent exists in the clinic today.
Thank you. So I hope the audience, I hope you're getting a sense of the vast sweep here. There are quite a number of brain programs around the world. They take different approaches, some more translational, some looking more fundamentally at the brain. We're making a lot of large-scale recordings, a lot of data flowing in. There are new tools that we're using to manipulate the brain. I heard the word optogenetics, which is the use of light to switch genes on and off. I don't know if that was clear to everybody. There are many labs involved, and we're still trying to get a picture of the whole brain, different cells involved, theory, and we're looking externally and internally with different ways we stimulate or measure. I wonder if at this point, I'm happy to ask questions all day, but I thought I might just take a quick temperature. If anybody in the audience would like to ask a question, I think there's a microphone right there in the room. If anybody has an immediate question for any of the folks on the panel. Yep, here's one right over here. Could I just ask you to say your name and where you're from when you ask the question, please?
My name is Kamran, and I am from JS Global, one of the largest securities companies in Pakistan. My question, because it's a topic related to brain, so there is a child in the family, and when she was born, at that time the oxygen was not proper, so she had dilation on the left side. Probably that condition is called as hemoparesis, and in that condition the motoring skills are weak, and the right side of the body is affected. Just wanted to know how science has improved in order to bring some cure to such kids as they're growing, and what can be done in that field, and what can be the possible things that can be done to bring that child at par with the other kids. Though we are told that it's minor, but how it has progressed and what can be done and what can be the solution going forward for such things.
I don't know if we have anybody who feels comfortable answering that question. It's not their direct areas. Is anybody?
So I didn't get it. So the case is the half brain is removed or something? I didn't get it.
Yes, the dilation on the brain that has resulted in the body having 50% movement in the right side.
Okay.
So technologically and otherwise, have we advanced so much that we can go and in the future we can look for some possible cures to such things like stem cells? That's I hear and read. Yeah. That can be the possible way forward.
So if the big part is removed from the brain, it's hard to use, in this state, use stem cell to reconstruct. But the brain is very flexible, so there's capability for the plasticity, which is, I remember this one case, is a case, her left brain is removed, but she wants to be the parallel Olympic player. Then she tried very hard to do the swimming. Then later he can move a little of the right side of the body. So in his left brain part, then this rebuild some connection to can partially control the right part of the body. So there's amazing capability of the brain to reconstruct the function. But if you remove too much of the part, in this stage it's very hard. Maybe in the future or in this stage, in this century, we can use this brain-machine interface technology to help you move your another part of the body. So you see like some EEG, some signal from the brain. If you want to do something, then you move the machine-built part of the body. So that's maybe one solution to that.
I think that one of the promises, I'm sorry I cannot provide help to you, but one of the promises in brain research and brain development is to tap into maybe one of the most significant findings of brain research of the last few decades is just how interconnected the brain really is and how plastic it is, the brain plasticity. So the brain is capable of rewiring itself in a variety of different ways. How to manipulate it, how to control it, how to cause it to happen, that's still yet to be seen, but it provides hopes for the future if we will continue with this type of research.
The question about stem cells, makes me think, Professor Okano. So we have a good deal of genetic information, but it's not always clear how to use that to benefit patients in the brain. Is there? But I know you're working with stem cells and other research. What can we say about that at this point?
Okay. So many diseases are caused by both genetics and also environment, and so takes Alzheimer's disease as an example, some familiar cases and sporadic cases. But even the sporadic cases, both genetic and environment plays very important roles. It is said that about 50% of the Alzheimer's disease genetics is very important. But different from familial Parkinson's disease, the genes involved not always clear in the sporadic diseases. So in order to deal with that, it's important. So patient-specific stem cells play very important roles. You can, by using patient blood cells or skin fibroblasts, you can generate iPS cells, and you can induce them into brain cells, neuron or neuron-associated and oligodendrocyte, or the brain-like structure called brain organoid that recapturates some cellular organization of the brain in a dish. And so, and by using that system, we can detect some disease-related phenotype in terms of the impaired synaptic transmissions, impaired neurite extension, impaired cellular migration, impaired mitochondrial activity, or impaired intracellular trafficking. That is, we can tell. So disease-related phenotype. And so then we can make the drug screening, potential drug screening that prevent the appearance of such a disease-related phenotype in a dish. And then we can have the potential drugs could treat those disorders, even if you don't know the genetic basis for that. That's one of the strategies. But so the problem is that sporadic cases, so genetic basis very variable from patient to patient. So we need lots of the patient-derived samples, and we should identify the most the drug that could affect most population, many population of the patient. So in each case, every drug should have responder and a non-responder, and we should be aware which drug could be involved in which kind of patient. Those patient-specific patient-specific approaches by stem cell would enable us to the new drug that treats those unknown genetic bases.
I see another question here.
Thank you. Just to follow up on this, I mean, what's the relationship between the microbiome and brain activity? And what treatments potentially instead of genetic treatments are there on the horizon? If anybody would like to comment, please.
Microbiome and Professor Okano.
Actually, in your gut, so huge number of the microbiomes living together with us, and maybe billions of cells. And so they are very, so it differs from person to person, So we can now determine what kind of the microbiome you live in your gut. by the sequencing ribosomal RNA. So each microbiome is known to produce different metabolite. Some of the metabolites, such as serotonin or neurotransmitter and like substances, or the particularly the lipid. Some lipid is very membrane permeable. It's known to affect the brain function, penetrating through blood-brain barriers. Those lipids produced in particular subset of the microbiome is very important roles in the development of the various mental disorders or keeping your brain in healthy conditions. That's why characterizing your microbiome patterns is very important. And so it's quite different person to person, but it also becomes changed according to some, if you have some dietary issues, and so it changes. So it's important to have what's your healthy condition in terms of microbiome. And the other issue is that which microbiome produce what kind of metabolite. So you can change in the bad condition into the good conditions in the monitoring your microbiome. That's why the microbiome can affect your brain function and disease state.
Can I just do a quick temperature check? Does everybody here know what microbiome means? I should have asked that at the beginning. You all comfortable with that word, so we don't need to define it? I think I saw another question over here.
Hi, I'm Jenny from KAIST, and I'm joining the WEF as a young scientist. So I work in neuroengineering, so I know a little bit of neuroscience, but not a scientist. And I was wondering, you probably do a lot of brain science in the mouse and in a small animal levels. And what are the challenges to move from this animal level to human? And how do you overcome those challenges?
Would you like to speak to that?
Yeah, this is a wonderful question. Okay. In fact, if under the microscope, we couldn't discriminate the cell from mouse or from human. So they were very similar in the cell shape. And also in the molecular level, also they share a lot of common things. So that's why. And also for genetically, in mice, we can find like more than 98% of human gene similarly. So that's why the neuroscientists or other field in biomedical use mouse as the major animal model to investigate the mechanism of the disease, to investigate a lot of principal function of the cells. And also use this mouse model, the scientists knew a lot of things about the learning, the memory, how they build this learning signal pathway and so on. And also the mouse model is very amazing progress recently. So there's one team led by MIT. They use Alzheimer mouse model. They found they use the 40 hertz light click. So you don't need to trade with the drug and so on. Even with the light, with the 40 hertz, in every 1 second, there's a 40 times flick of the light. Then they can reduce beta amyloid, which is one of the major pathogens of the Alzheimer in mouse model. So they are now trying to use this new discovery to try whether they can help the human. And in fact, there are a lot of effort to use mouse model to find a new drug. They failed. More than 99%. They take like millions, billions of dollars. But at the end, when they use the clinical trial, in fact, it's failed. But still, for the basic level, to investigate the mechanism of the disease or something like related to cognitive function, mouse model contribute a lot. So I think a lot of neuroscientists, we are very appreciative of mouse. They are contributed a life for a whole human being. Okay.
Yeah, I think one of the big surprises in the last 10 years in neuroscience research is that mice are actually much cleverer than we thought and actually can perform quite sophisticated decision-making tasks. And actually for the International Brain Lab, this new initiative that I'm involved in, We're planning to use a task that's based on foraging in the mouse, which is an innate behavior in the mouse. It's vital for survival. It has obvious counterparts in humans. Humans also do kind of foraging in many of their behaviors. And it's a very rich behavior because it requires processing sensory information with focused attention, reaching decisions, acting, learning from the results of your actions. So it's very multidimensional, involves interactions with multiple brain areas, and provides a rich platform for then making connections from what we learn in the mouse to human behavior.
And also, I will add one more. So the mouse not only is a very good genetic model for human disorder and memory and learning, and also for some environmental factors, which is we are puzzling to cause some problem like depression, anxiety. Like there's one phenomenon. So the survivor after the earthquake, which is they are under the rub, then they get rescued. But this group of people is more easily to develop to depression and so on. Then we use a mouse to mimic that condition, put them in a restrained area in the tube. Then only 1 day later, this mouse, after 1 month, they develop to some depressive-like behavior. And also in their brain, the neurogenesis is reduced and so on. After you treat them by the antidepressant, then they can rescue this problem. So mouse model is very useful for human beings, for neuroscientists to deal with a lot of puzzle. Okay.
So, did you have
If I may
comment?
add a question or just an anecdote about that. At the end of the day, there are 420 molecules that were tried on mice successfully on Alzheimer's disease, but all of them failed phase 3 clinical studies in humans. So it's some food for thought. What are we missing? So I don't know if there's an answer to it, but that's the fact.
Yeah, okay. Yeah, so in terms of mouse model, it's provide excellent model for studies function of the molecule and cells, and also a very elementary action of the how the synaptic transmission occurs, and also innate behavior. But as said, so there's some gap between the human and mouse. We should realize that part. And for example, so the brain region called prefrontal cortex, some part is missing in the rodent. So in order to detect the prefrontal cortex and driven the working memory and so higher brain functions, obviously, in addition to mouse, so studying a non-human primate is very important. And so the difference mouse and rodent is it's 3 points. And genetic information, the lifespan is very, short and 2 years old, but we live at 70 or 80 years old. It is sometimes difficult to recapture late onset diseases. 2 years could be too short to recapture it. And also the brain structure is different. So on the purpose you are interested, you should use most appropriate model system. Mouse has much advantages in terms of the well-developed molecular genetics, but so when it's not it's perfect, we should use other model. And also human cells is very important because some microRNAs and non-coding RNA and epigenetics are different from mouse and rodent. That's why we are very keen to use human iPS cells to the drug screen, not mouse cells. But so we could easily test its in vivo effect. So in many cases, combination as a mouse, non-human primate, and finally in the human. So in your brain minds, brain initiative, so cross-species comparison is very, very important in terms of brain structure, connectivity. So that's it. Mouse model is great, but not always perfect. So they should be complemented, but many other techniques and many other model systems. That's I think.
Come back to the questions here in just a minute, but I was just thinking, Professor Allen, what are we missing here in this picture of disease model and treatment?
Oh, if I could just add on to the mouse
do.
human thing first. There's been some interesting studies actually showing that human astrocytes, these cells that touch all of the synapses, are actually quite specialized and have quite a few differences in human compared to rodent. So for example, the human versions are much larger than the rodent counterparts, so they have an ability to be interacting with and sensing many more neurons than a rodent one would. And they also have some different shapes and different processes that actually span the whole region of the brain instead of being localized. We have no idea what they're for. So I think this idea of we can understand a lot in the mouse about the general cell biology, how these cells interact, but to get to the real specialties and kind of humanness, then we have to be looking in the right place.
I saw a question back here. It's right behind you.
My name is Ishwar Prasad. I'm an economist. I was intrigued that many of the issues that you've been talking about sound to me like they're sort of experimental in nature or in the way you approach them. But there was a mention about theory, and it was also implicit in your remarks. I'm curious about whether there is really a theory of the brain you're working towards, and can we think about it in the standard form of a theoretical framework that sort of generates testable predictions? And if there is, in fact, some sort of theoretical framework, do all of you here agree on it, or is there a lot of dissension about what the theoretical framework is? And more fundamentally, do you even need a theory in order to do the sort of work you're doing? Do you let it guide your empirical work?
Yeah, I think that's a really fantastic question. And actually, I think we actually lack a general unified theory of how the brain works and how different brain areas interact to generate behavior. And that's for two main reasons. First of all, it's a really, really tough problem, obviously. And secondly, the experimental and theoretical communities have been working pretty much in parallel and often quite independently. Often it's because they speak a different language from each other. And so that's why it's so important for us to integrate experiment and theory in new ways, and in particular to be able to embed theorists in experimental labs and vice versa, so these two communities can learn the same language and talk to each other more efficiently. And this is part of the motivation for this new collaboration project that I've been describing. So we hope that actually by providing new links between experimentalists and theorists that, first of all, will give theorists access to the best kinds of data in the best experimental labs, people who are collecting the terabytes of data from the new large-scale recording techniques that are emerging around the world. And also they'll be able to actually work closely together with the experimentalists to devise experimentally testable predictions to help validate or falsify these theories. So it's an absolutely critical point in neuroscience.
I saw a lot of nodding. Do you agree, Professor Okano?
Yeah, okay. So, yeah, I agree. This is very important questions. As he said, so now we still don't know how our brain works in the coordinated. It is due to the lack of the complete map, how the brain is wired to cell-to-cell communication. So without those knowledges, we can tell how they are interconnected to each other. That we could not make the theory. So unbiased data is very important to have a complete map. If you think about our cellular functions, the whole genome sequencing data was very, very useful to understand how our cells behave and which genes are expressed. And now we can predict the cellular function to some extent. So that's why, in order to understand how our brain is coordinated, it's organized, we need a map. If we could have a map, we could make the simulation, the brain the information process. And maybe at that stage, maybe artificial intelligence, human brain like AI, will be important to understand how our brain is organized and coordinated. For that purpose, we need a map, complete map. We are very initial state.
Been waiting patiently here.
Thank you. My name's Tim Verstein, and I'm a cognitive neuroscientist. And I'm very sympathetic with these big brain initiatives that are being put forward. But our recent experience with the Human Brain Project in particular in Europe showed that our ambitions aren't being matched by the kind of scope of these funds that are being put forward. So, Henry Markham promised a biologically realistic simulation of the entire human brain cell for cell. And for $10 billion, what we got was a 1 cubic millimeter simulation of a mouse somatosensory cortex. So I'm kind of curious what the panel thinks would be realistic benchmarks for evaluating the success of these big brain initiatives, given that the scope of coming up with a comprehensive simulation or model of the full human brain is an unrealistically big problem given where we are today.
I think that's a very important question, and I think the lessons that we've learned from other big initiatives in different kinds of science, for example, in physics or molecular biology, is that these big collaborations really only work well when they're targeting a very focused, well-defined goal, right? Like sequencing the human genome, finding the Higgs, for example. And I think it's therefore important for these international initiatives to really clearly frame what success is going to look like. So for our part, in our collaboration, this International Brain Lab, we have a very common, well-defined goal, which is to understand a single brain function, to understand this foraging behavior in the mouse. And we want to achieve this by having unprecedented interaction between experimentalists and theorists, as I've explained before, and also unprecedented sharing between labs, sharing data, sharing setups, sharing analysis tools, etc. And we think that only in this way that we can achieve these big goals that we set for ourselves.
Maybe.
Actually, scientists are making theory. If we could know the whole entire map, can we predict the animals' total behavior or innate behavior, etc.? So now we have a whole map for the C. elegans, a very simple animal, and try to now the Drosophila maybe soon. So if the total entire map is elucidated, now maybe we could have the technology or method to predict the behavior and neural functions. It's very important. So how our brain function is simulated using the simple animal models. Then we can it can jump up to the more complex in the animals. So step by step is very important. That's why Henry Markham now working the mouse. But so in the future, we'll be able to know that entire human map in some certain levels, macro level, meso level, and micro levels. Which type of the information is important to understand the behavior and brain function. And so using a more simpler system is very important at this point. In the future, by using those technology, we'll know how our human brains works. We need to explain the same issue to even the politician.
So I think that a major part of these initiatives are focused on basic science and understanding the brain in a more comprehensive way. Maybe we could think of initiatives, and there are some initiatives that are thinking about a key performance indicator of the project, if you will, is how many patients we can help. Next year, 5 years from now, 10 years from now, and 20 years from now. So a lot of these projects are very far-reaching. They're looking ahead many, many years. But there are things that we can do even today, and I will echo what you just said, that we don't necessarily have to know the theory in order to help patients. And an interesting anecdote that I just read recently about Tom Insel, the previous director of the NIMH, was one of the panels that he participated. One person asking the following question, said, the house is on fire and you trying to figure out the chemistry of the paint. So it's important to figure out the chemistry of pain, but is there anything we can do in order to distinguish the fire sooner rather than later? One of the key areas that I think we should discuss and consider is that everybody talks about how big data is going to transform healthcare and whether there is a role in neuroscience for that to really change paradigms in the way we prognose, we predict diseases, predict behavior, even without knowing the theory, or predicting which treatment will work and which not, even without knowing the theories yet, but at least bring some clinical tools that can alleviate. So this is an area that we are focusing heavily on, collecting a lot of data and trying to see how the different parts relate to each other, how electrophysiology relate to structure, relate to behavior, and try to, with big data and machine learning, to try to come with prognostic values to see whether a treatment is going to work or not for depression, or predict a relapse, or see early Parkinson's disease. So these kind of things, I think, will transform our knowledge about brain in the next few years, the big data and artificial intelligence.
I think this is a good pivot point, actually, to start to look ahead a bit about some of the things we're maybe anticipating in our areas of research and things that maybe represent challenges that are still need to be surmounted. Professor Allen, could I ask you to think forward a little bit in the areas of the cells that you were talking to us about, which I would like to hear more about. What are some of the areas that you're finding really promising that are being explored now as far as brain?
Yeah, so I think we talked a little bit earlier about plasticity of the brain. So the ability a brain has to really remodel itself in response to challenges or experiences. And I think what we don't really touch on is this is something that's much more powerful in the young brain and tends to decline as we become adults and as we age. So one of the reasons that we think these glial cells can be very important in these processes is experiments were done 30, 40 years ago now, where they took young glia, young astrocytes, and put them in an old brain and showed that was able to reinduce this plasticity, this strong ability of neurons to remodel. Whereas if they took old astrocytes into an old brain, it had no effect. So what that tells us is there's something that the neurons are in an environment in the old brain that's stopping them from being able to respond to experience or remodel, and that these glial cells they interact with are perhaps the gatekeepers to this, and that they're kind of stopping this happening in an adult brain. So now with the new technologies we have for sequencing and determining how these cells function in the young and the old brain and what the genes they express, like we talked about before, we think perhaps we can get some insights into understanding what's special about these cells in a young brain that we could then perhaps try and reintroduce into an old brain or an injury or disease in order to encourage better neuronal function and recovery in different disease situations. So I think that's one area where we can think about understanding how these other cells in the brain normally function to normally help you in development, and how we could repurpose that for different diseases and help in treatment of these disorders we've been talking about.
So, we've thrown so many ideas in the air. I'm thinking as, with an eye to time. We have just a few minutes left. We've talked in the last 10, 15 minutes or so about stem cells in the brain and what we can do there, the benefits of using mouse models in different ways to kind of probe more about the brain, that we maybe lack a complete theoretical understanding, and yet we can still learn many things. That we need a map, and we have a lot of work there, and that our ambitions are rather vast. As we sort of close toward the end of this session, I'm thinking, what would each of you like people to take away from the areas that you're working on? What is one thing, maybe Professor Lee, we can start with you. What is one thing you would like people to know about your area or to keep in mind?
So as a neuroscientist, I have a confidence for two things. One is use the new technology to investigate the principle of function of brain. A lot of people in the field will agree that Carl Dossel from Stanford, he discovered this optogenetic, gave us a great tool because the brain is so complex. But use his tool, and we can dig with what's the circuits of the brain to deal with the fear, the love, and also something difference. And also, the laboratory uses this kind of technology can make an animal, like when they smell the disgusting things, they can think it's a very pleasant things. So this already happened in the field. So using this technology, we will in the future can figure out a lot of function of the brain, basic brain function. And another thing is the neuroscientists work together all over the world to make a progress for the cure the disease of brain. So for the mental disorder or the neurological disorder, I also have content for that. But one thing, as a neuroscientist, I have a little of the fear is the evolution of AI. So as everybody knows, AI is this concept started from last century, but only less than 1 century, they can hear and understand what you're saying. They can discriminate the imaging and so on. So this is some of the basic function AI already got it. And the human brain is started from the animal brain. So probably already evolved for millions and millions of years. But the human at this stage, they may not beat with the Beethoven to make the compose the great music or the great master of paint and so on. So the human brain developed very slow. So there is one clue, maybe the nature left for us. There's some of people with superhuman power or brain function, like they have a great memory skill or the math skill, something like that. If less neuroscientists work on that, maybe we can make a more effort on this kind of potential function of the brain, so accelerate the brain evolution. And another resolution is build this brain-machine interface. Like in the future, if we want to beat AI, maybe there's a hybrid of human and machine. So that's what I thought.
Professor Hausser, any quick thoughts?
Sure.
Sorry.
So I think that to make transformative leaps in our understanding of the brain, we need to change the way that we do neuroscience. So we can't remain a cottage industry forever, in which we have individual PIs working in isolation. We need to get organized and find new ways to collaborate and share tools and share data. And this is going to go against the grain of the common culture in neuroscience. And it means we actually have to create a kind of a cultural transformation in the field. And there's an African proverb: if you want to go fast, go alone. And if you want to go far, you go together. And actually, what we think is that by collaborating, we can actually go both faster and further than any individual lab can, because it's going to be much more efficient, and it's also going to be much more fun.
I think actually that feels like a really great note to end, to go farther together, to go faster together, while we strive to understand this amazing piece of what we're in our skulls. I want to thank everybody for a really wonderful and stimulating conversation. Can we please thank our panelists here? Thank you.